Hello, welcome back to Pure Science Education. Today you will be looking at mitosis and meiosis. So what is the difference between these two procedures and why are they so important? So as you can see in the diagram, we have mitosis that causes the splitting by cell division and results in two identical daughter cells. And if you look at meiosis, by cell division, it forms 4 non-identical daughter cells. So let's look at how that occurs. So for a start, let's look at the differences between mitosis and meiosis. So mitosis, the way I remember, will be head to toe. That means it makes or repairs body cells from head to toe. And if you look at meiosis, it makes sex cells, which in science we'll call gametes, and male will make sperm and female will have eggs. So let's take it to the next stage. So in humans, we have diploid cells. Diploid means double number of cells because you get 23 chromosomes from mum, and you get 23 chromosomes from dad. So as humans, we have a total of 46 chromosomes. So let's look at each of these individually. So if I was to start with a chromosome, if I start with a single pair, that is called a centromere. And if I was to then make a diploid, duplicate this, then I'll get something like that. And this is duplicate chromosomes making 46 pair. If the chromosome is still attached in the center by the centromere. But we can say that this is 46 chromosomes, even though we have doubled up, although we could say it's 92 chromatides. Okay, so let's look at the phases. Now the first phase we're going to talk about is interphase. Now interphase does not take part in mitosis or meiosis, so does not take place in mitosis or meiosis, but it is important as it causes duplication of chromosome. So at this point, we're now going to start looking at mitosis and meiosis, and we're doing in parallel. So to learn mitosis and meiosis, if you sit in your exam, the best way to learn it is something called PMAT. So if you can remember the PMAT, you'll be able to do your mitosis and meiosis in your exam. So P stands for prophase. And if we was to look at the prophase in the mitosis, so if we do mitosis first, and then chromosomes are condensing, getting thicker. And if I was to do the same for meiosis, because meiosis happens twice, I'm going to write P1, A1, T1, and P2, M2, A2, and T2. This occurs twice, so we're going to write out twice. Now, if you was to look at this side for the prophase, we're still doing the prophase, and because it's going to duplicate, so basically you'll have the same gene for both sides. Let's just draw that again. So say for eye color and so on, but because it's a one gene, it will be the same size and have the same genetic information, but what it'll do, it will make up and join with the homologous pair. The reason being because one pair will come from mum, one pair from mum, and one pair from dad. So you ended up getting that, and then you might have a cross section as well. Let's look at the M. So the M stands for metaphase. Now the way to remember M is middle. The reason being M is middle because all the chromosomes align in the middle, as the nuclear membrane already has been broken in the previous stage. So chromosomes align in the middle. Like I said, the nuclear envelope has already been broken, and as we used to look at the mitosis, metaphase, then if I just use for chromosomes and for this explanation, then we'll end up with 1, 2, 3, and 4, and you will see that they all are aligned in the middle, so like that. And if you was to do it for meiosis, you will see that if you was to do the same thing, but because they come in pairs, they're still aligned, and again aligned in their pairs. So they go in the individual pairs. The M1 is pairing up in the middle, and they're not in a single line because they have got a pair. Right, so let's move on to our A in the maths. And this time A stands for anaphase, and again to remember A, you can think of away, which is an easy way to remember this again. And this time what we're doing is our chromosomes, the chromatides are pulled away by the spindle, so they move into the opposite side of the cells, so you end up with your so if I just do the 4 pairs and this side, so what you'll see, they have moved and been moved to opposite sides. So chromatide are pulled away by spindles. So that's where, and this is for the mitosis. And if I was to then look at the A1 for meiosis, so A1 in meiosis, you will see that, I'll do the pairs again, that they are gone in the pairs and they've been pulled, the A1, its chromosomes are moved away, but not the chromatides. So let's look at the T. T is telophase, and in this phase you end up making your final stage where the cytokines help to make up your final phase, where the mitosis and meiosis comes to the end. And in the end, if you're doing mitosis, you end up with, you get 2 diploid cells, and they're used to repair or make new cells. So in this case, you're going to get 2 daughter cells, and they're going to be identical. So identical daughter cells, and they are used to repair things in the body, so hence they're identical. And if you just look at the final stage of meiosis, because it is sex cells, in male you make sperm and female you will make eggs, and they are haploids. That means they got half the number of cells. So if I was to do the same thing, and you end up with, let's just draw it slightly differently. So these are your half number of cells. Here you got identical cells. Here you got half the number of cells and the non-identical. So you got 4 non-identical cells. And that's a quick whiz through your mitosis and meiosis.